Good morning, good afternoon, good evening, depending on where you are. My name is Jean de Dieu Nsenganeza. I'm from the University of Rwanda, Center of Excellence in Biodiversity and Natural Resources Management at the University of Rwanda. Today, I'm going to share with you the work I have been doing and also still doing on that, which is called distribution of flies across altitudinal gradient in Nyungwe National Park in the south and west of Rwanda. This part of the presentation is made of one part of my master's thesis and another part of the Diptera collection initiative that we started in Rwanda. As introduction, the Diptera flies, they play a central role in ecosystem services and economic value. They support reproduction of crops, and also they are good pollinators. We sampled in Nyungwe National Park, which is UNESCO heritage site. And it is known by its biodiversity. It hosts more than 1,100 plant species, 85 mammals, 13 primates, and more than 300 bird species. And it supports a diverse of amphibian, reptile, and butterflies. So this work has aim of documenting diversity and distribution of flies across altitudinal gradient in Nyungwe National Park. So we wanted to examine how flies are distributed in Nyungwe National Park, since Nyungwe National Park is made of different hills. Because other taxa were documented, such as butterflies, amphibians, small mammals, birds. So I want to take this initiative to start working on the flies. So I wanted, my specific objective was to assess the Diptera of Nyungwe National Park as preliminary checklist to know the distribution along elevation gradient, and also to evaluate the effect of environmental parameters such as temperature, humidity, canopy, vegetation type. Here is the study site. This left part is the map of Rwanda, which is a landlocked country. And this is green part is Nyungwe National Park, where we sampled. And the green part is the sampling station. We collected the sample in 6 sites located in Nyungwe National Park. And those sites they have different height. Since we were measuring the altitudinal gradient, we choose those sites because some are at the highest elevation, while others they are at the lowest elevation of the park. So here on the left column is the number of the sampling station we made at each site, and it will depending on the height of each site. So now we categorize those elevation into low, medium, and height as it is indicated below. And then we have sample the Diptera by using sweep net, pan traps. And pan traps, we used 3 different color of pan traps. yellow, blue, and white, since those colors are more are attracting flies. After that, we also measure environmental parameters by using GPS for getting the elevation gradient, thermohygrometer for temperature and humidity. After that, we calculate family richness, linear regression model, and non-metric dimension scaling to measure the effect of those environmental parameters on distribution of those flies. Then, as the result, we got a total of 691 individuals in all sites, which has been distributed into 36 families, and where Muside was the highest dominant, followed by Tachynidae, Syrphide, Califoride, Chloropide, and Pholidae. Other families were represented also with 1% or less. Then we have also measured the elevation range, and we counted the number of the families recorded at each elevation range. We recorded 35 families in the middle elevation, 21 families at the low elevation, 15 families at the highest elevation. So at the both elevation ranges, Muside, Tachynidae, Syrphide were the most dominant. Now we made a simple linear regression model to see the effect of elevation on the distribution of those flies, where we found there are significance difference because we got p-value which is less than 0.5. Here the p-value is 0.04, and then the result indicates also positive correlation, because if you can see, there is this regression line is looking upward, which indicates positive correlation. And we have seen high concentration of family in the middle of elevation, as it is indicated by this cycle. After that, we have also look for the temperature, and we have categorized low temperature, middle temperature, high temperature, as it is indicated down here. And then we have seen that there is no statistically difference since p-value is greater than 0.5. And we found negative correlation because the linear regression is going downward. Now, about the canopy, we categorize the canopy in 3 ways: open canopy, semi-open canopy, and closed canopy. And you have seen that in open canopy is where there are more family richness compared to other canopy type. About the vegetation types, we collected in fans and primary forest. Those type of vegetation were depending on where the sampling point were randomly fallen. So there was no significant difference, since the number of family at both vegetation type are equal to 28. Now, after that, we have calculated nanometric multidimensional scaling to see the effect of all environmental factors on distribution of those flies. Here, I drop out an example about this map, this figure, which illustrate, like, for instance, uplift cluster, the family like Glossinidae, Scatophagidae, Stratiomidae is linked to higher canopy and higher humidity and also higher elevation, as it is indicated in this map. Now, this is an example. Now you can now able to interpret the remaining family according to the examples I demonstrated here. Now, about the discussion, the distribution of flies seems to follow distinct and model pattern. And the microclimate factor at the middle elevation favor most Diptera community. And because those microclimates, it favor the survival of Diptera larvae. And the high species was also found in semi-canopy. In semi-canopy, it was heterogeneous, which has access to light and also flowering plant. The higher number of the Diptera family was recorded in cooler area, which is between 14 degrees Celsius and 21 degrees Celsius, except for generalist Diptera. So now the future direction. So far we have specimen. We need to make metadata are available. Now we need to make DNA study. So we have to start taxonomic study and to develop identification keys to the species level. All those Diptera were identified to the family level by using Afrotropical Diptera volume 3. So now we need to develop identification keys to the species level. After that, we start initiative of collecting for the Diptera collection. So all those specimens collected in Nyungwe National Park, 15% were identified to the family level, and 75% of all collected specimens are now mounted, and And then here is our infant stage, if I can say so. We have some boxes which we are working on, so that we are collecting to see if we will fill the Diptera specimen in all those boxes. So we are at the infant stage, and we hope we are going to grow this collection as it is our wishes. So I'm thankful. I'm acknowledging the University of Rwanda African Museum, who supported the fieldwork Center of Excellence in Biodiversity and Natural Resources Management, which is a UNESCO Category 2 center, and also African Park. I thank you very much for your kind attention. Here is my email address. If you have any question, any concern, any advice that you can provide, please feel free to check out to me, and then I will be grateful